Hello, it's Arkasha from MCAT Simplified. So today we're going to be discussing amino acids. So in the body, there are 20 standard proteinogenic amino acids. And what I mean by that is that these amino acids are used to synthesize proteins, hence proteinogenic. Furthermore, out of those 20, 9 of those amino acids have to be obtained from the diet. So they are essential. And the remaining can be synthesized in the body. Now, all amino acids share a common structure, okay? So all of them have the central carbon C, and that central carbon is attached to 4 different groups. The first is this carboxyl group in the red. Then they're also attached to this amino group, and then a hydrogen. And then what gives each amino acid its unique chemical and physical properties is this R group, which is the side chain, okay? Now, before we actually dive into the individual amino acids, let's talk about some general properties. So for one thing, in eukaryotes, amino acids are typically found in the L configuration. And what I mean by that is that when these amino acids are drawn thusly, the highest molecular weight atom attached to the central carbon, which is the nitrogen here, appears on the left side of the page, okay? That's the L configuration. Now, this is in stark contrast to how many carbohydrates are found, which are usually in the D configuration. And that's an important tidbit to remember for the MCAT. Now, additionally, amino acids, they can be charged or they can be neutral. And we'll see that when we actually look at the individual amino acids. But importantly, even within the backbone itself, this carboxyl group can be deprotonated and form an ionic negative charge. And then this amino group here can actually accept a protein and become positively charged. Okay, so the backbone can also be neutral, positive, or negative. Now, additionally, this imparts upon amino acids an interesting property, which is that they can form these Zwitter ions, okay? And that's a German word, and it essentially means a molecule that can have both a positive and negative charge in a way where the charges cancel out and the molecule overall is neutral, okay? Additionally, these ionic charges, they influence the properties of these amino acids and also the proteins that use these amino acids to be synthesized. Okay, so of course, with ionic charges, there can be electrostatic interactions of various sorts, and that influences both how the protein is folded when it's synthesized from amino acids, as well as how these proteins can interact with other molecules. Now, amongst the amino acid types, these are usually categorized into 5 different categories, all right? So there's nonpolar amino acids, there's polar amino acids, and then there's aromatic amino acids, which also share properties of polar and nonpolar amino acids. And then there's negatively charged and positively charged amino acids. Now, amongst these categories, some general things to know, especially for the MCAT, is that nonpolar amino acids are typically found in the inside of proteins. So as a protein folds, because it's typically found in an aqueous environment, so there's water molecules around, the protein will fold in such a way that the hydrophobic, the water sort of hating or disliking amino acids appear in the center of the protein so that they're shielded by the more hydrophilic, polar, and ionized amino acids on the outside. That way, it minimizes energy, okay? And if you want to know more about that, go check out our videos on proteins and protein folding, okay? Now, amongst the polar and charged amino acids, these are usually found on the outside of proteins, as I just suggested. And then also, for both nonpolar and polar, when we're talking about proteins that appear in membranes, the nonpolar ones will usually form the intramembrane domain of such proteins. And that's because, of course, the inside of membranes are lipids, and lipids are hydrophobic, okay? And so because like attracts like, if you have a protein inserted into this membrane where the inside is hydrophobic, you also want similarly hydrophobic amino acids of the protein to be on the inside. And therefore, the intramembrane domain of membrane proteins are usually consistent primarily of nonpolar amino acids. And then inversely, the extramembrane components, which are usually the ones that allow these proteins to interact with other proteins outside the cell or inside the cell, those are usually polar amino acids and charged amino acids, all right? And additionally, the polar and charged amino acids, those are the ones that predominantly play a role in various enzymatic catalytic mechanisms, all right? And then lastly, aromatic amino acids. An important thing to know about these for the MCAT is that they absorb UV light, okay? And this can actually be used in various protein analysis techniques and molecular analysis techniques to detect proteins and also just to determine how many amino acids, relatively speaking, are in a solution, okay? Now, one last point is we mentioned essential amino acids, okay? And there's typically 9 essential amino acids that have to be obtained from the diet. And so a useful mnemonic to memorize this is PVT Tim Hill. So the P here is phenylalanine, okay? That stands for phenylalanine. And V is valine, T is tryptophan, the second T is threonine, okay? The I is isoleucine, the M is methionine, the H is histidine. And then this I is just there to make the mnemonic nice. It doesn't stand for any amino acid. And then lastly, the 2 L's are going to be lysine and leucine. And so that's PVT Tim Hill. That's an easy way to remember it. Okay, so now let's talk about the different groups of amino acids and which amino acids belong to them. All right, so first we're going to have the nonpolar amino acids. And the first amongst these is perhaps the simplest, which is glycine. And notice that here in the parentheses after each name, I'm going to have the 3-letter abbreviation and then the 1-letter abbreviation. And it's super important that you guys are able to quickly interchange them and recognize which 1-letter abbreviation corresponds to what amino acids. Okay? Now, notice that also here in the red for nonpolar amino acids, we're going to have the side chains. So the backbone, like we said, is always the same for every amino acid, but the side chain is what makes them unique. Now notice that glycine is just an H side chain, just one hydrogen. And also notice that that central carbon is therefore attached to 2 hydrogens and then the amino and the carboxyl group. So there's not 4 unique constituents. And that actually makes that carbon achiral. And incidentally, this is the only amino acid of the 20 we're talking about that is achiral. So it's not optically active. Okay. Now, additionally, it's a very small amino acid with a very small side chain that is very flexible. All right. And that's an important physical property that's utilized in the folding of proteins. And it really helps to dictate a lot of protein structure because this allows, as the protein folds, for this amino acid to be positioned in areas of very tight turns. Like, for instance, in beta-pleated sheets where they have very tight turns. Glycine would you could predict, in other words, that glycine could be found in those locations. However, in other secondary structures like alpha helices, if this glycine is found within the helix itself, that can be detrimental and that can actually break the helix down. Okay, it can destabilize it. Now, additionally, we have alanine. So that's ALA or A, quite simple to remember that. And this is just a simple methyl side chain, really. Nothing really too special about that one. Then we have valine. And so valine is just V, nice and simple. This is one of the essential amino acids, right? Remember PVT, Tim Hill, okay? And then this one has this side chain of a CH group attached to 2 methyl groups, okay? So it's kind of like a little Y shape. And then we have leucine. So leucine has L, okay, as the abbreviation. And this one has a similar side chain to valine, except it has an additional carbon. Okay, so it goes methylene group, this CH group, and then 2 methyl groups. So it's just the valine chain extended by 1 carbon, effectively. And this happens to be an isomer of another, a constitutional isomer of another amino acid that we're going to talk about called isoleucine, and you'll see that in a moment. And then additionally, this one is also essential. So then we have isoleucine, which has the 1-letter abbreviation I. And like I said, this is an isomer of leucine, and you can see that just like leucine, it has 4 carbons in the side chain, but in this case, the methyl group has moved from the 2nd carbon to this 1st carbon, all right? And notice also that this carbon here, this 1st carbon, is attached to 4 unique groups, right? This little chain here, this methyl group, this hydrogen, and then the alpha carbon. And so it's actually also a chiral carbon, all right? That's the beta carbon. The 1st carbon attached to the alpha carbon is the beta carbon. And in isoleucine, the beta carbon is also chiral, optically active. And like the previous 2, this one is also essential. Then we have a very cool looking amino acid, which is proline, P. And this one, as you can see, its side chain actually attaches to both the alpha carbon and the amino group in its backbone. All right, so it actually forms this ring. And as you might imagine, this ring is very, very rigid. It's not very flexible, unlike glycine. And again, this significantly can impact the structure of a protein, all right? Because it's very rigid, it can't be found just anywhere, and it certainly can't be found in these alpha helixes, okay? In fact, proline, if it was to be incorporated into the inner structure of an alpha helix, would destroy the helix. It would not be able to produce that helix if it was there, all right? And so that does significantly dictate the folding and the overall structure of a protein when proline is present, all right? Now, we also have this amino acid called methionine. So methionine is interesting. It has this CH2 group, and then it's attached to this sulfur atom, and then a methyl group at the end, all right? Now, this one is a really important amino acid because in eukaryotes, it's actually the amino acid that is encoded by the start codon, AUG. And by the way, this is a really important one to have memorized because it can come up in passages related to genetics and DNA replication, transcription, translation, the central dogma, okay? And this one is also an essential amino acid. And then lastly, amongst the nonpolar amino acids, an amino acid that can be sort of controversially classified as nonpolar, it's cysteine. So CYS or C, all right? And notice that the side chain here is a CH2 group and then the thiol group, SH. Now, this thiol group, it is actually slightly polar, but it's not polar enough to where it really significantly interacts with water molecules. So generally speaking, this can be classified as either polar or nonpolar, but if it's polar, it's only polar slightly at best. And since it minimally interacts with water molecules, it's not usually found on the surface of proteins like other polar amino acids. In fact, it would more likely be found on the inner core of a protein with the nonpolar hydrophobic amino acids. All right. Now, this is an extremely important amino acid for dictating and stabilizing the structure of proteins, and especially the tertiary structure. And that's because these thiol groups, if two cysteine amino acids or residues, when they're incorporated into a protein, amino acids can be called residues. When two of these are found close to each other in a protein, these two side chains, the side chain of one and the side chain of the other, can become oxidized. So in other words, they lose electrons, okay? And that creates a bond between the two sulfur atoms. And this is called a cysteine linkage. So this is a cysteine amino acid, and this is called a cysteine or cysteine linkage, okay? Also, and this whole group here is referred to as a disulfide link or a disulfide bond because there's two sulfurs, okay? And this is super important for stabilizing the tertiary structure of proteins and also quaternary structures. And this is a covalent bond. So unlike a lot of other intermolecular interactions that stabilize proteins and dictate the folding of proteins, this is one of those that is actually covalent. The other ones are typically non-covalent, like hydrogen bonding and electrostatic interactions of various kinds. And again, if you want to review that, we have a video on it, all right? So that's it for the non-polar amino acids. Now, also here, we have the aromatic amino acids. Now, the first one of these, and the reason they're aromatic is because you might notice that they have these special rings in the side chains, and these are called aromatic systems or aromatic rings, okay? Now, the first of these is phenylalanine. So phenylalanine has the one-letter abbreviation of F. Now, this is actually the first that is not abbreviated with the first letter of the name itself. So notice that we have proline, which has taken P, so we can't use P again, so we have to use F. Fortunately here, an easy way to remember this is that phenylalanine is pronounced, it starts with F, so phenylalanine. So it makes sense for it to be abbreviated as F, phenylalanine F, all right? And notice that the side chain is a CH2 group and then the benzene ring, the aromatic system. And this is an aromatic amino acid, but aromatic amino acids also happen to be hydrophobic. And the reason is because this is a hydrocarbon structure. It's just carbons and hydrogens. It's also hydrophobic, all right? So it shares properties similar to these amino acids here that we talked about. And phenylalanine, because it has this aromatic ring, just like the other two here, it can absorb light at a wavelength of 260 nanometers. So this is a wavelength, all right? And it is also an essential amino acid, okay? And there is a condition called phenylketonuria, which is basically when phenylalanine cannot be metabolized any further. And that means that this amino acid will actually accumulate, and if it accumulates too much, it can lead to toxicity, including neurotoxicity, and that's something you learn about in med school. Now, tyrosine is another aromatic amino acid. And notice that the abbreviation for it is Y. Okay, now this one is very weird. And really the first thing that you think about is, why though? Why is it a Y? And I think that makes it pretty easy to remember because, you think tyrosine and you're like, well, why is Y the abbreviation Y though? So I would remember it that way. Now this one has this OH group, but even though it has this polar moiety or polar functional group here, the overall molecule is actually still hydrophobic. But it does create for some potential interesting interactions since this OH group can serve as a hydrogen bond acceptor and a hydrogen bond donor. But overall, it is considered to be hydrophobic for the most part. Now, interestingly enough, tyrosine is not essential because it can be synthesized directly from phenylalanine, okay? And this molecule, tyrosine, is subsequently converted in the body, if it's not being used for protein synthesis, into thyroid hormone, which is an extremely important hormone for metabolic regulation, and then other types of hormones and neurotransmitters classified as catecholamines, and that includes epinephrine, norepinephrine, and dopamine. All right? And then lastly, we have this really cool looking amino acid called tryptophan. All right? And tryptophan, okay, it has a CH2 group, and then it has this double ring aromatic system with the two fused rings. Okay? And incidentally, this tryptophan molecule has the largest side chain. And honestly, that's a big win or a big dub for tryptophan. And so that's how I remember that tryptophan is denoted by W, okay? And then this one really absorbs UV light very strongly at 280 nanometers, okay, this wavelength. And it's really this amino acid that in proteins has sort of the strongest absorption. And typically, when you do these sorts of analysis experiments, you can detect proteins or like the relative amount of amino acids and stuff like that in solution by detecting absorption at 280 nanometers, and that's thanks to tryptophan. And that is an important fact to remember for the MCAT because there can be experimental passages about different experiments that they're doing, that they're proposing, where part of it is basically detecting proteins or amino acids using this wavelength of light. And you got to realize it's because of tryptophan, okay? And then also, kind of like how tyrosine is a precursor to hormones, so is tryptophan. Tryptophan is a precursor to serotonin, and then also melatonin, the sleep hormone. And then also it happens to be a precursor to vitamin B3, aka niacin. And tryptophan as well is also essential. So now let's discuss polar amino acids, and specifically the polar uncharged ones, the non-ionizable polar amino acids. So the first of these is serine, with the one-letter abbreviation S. Now, this one has a methylene group, CH2, and then this hydroxyl group attached to it. So it has a hydroxyl side chain, and of course that means it can hydrogen bond. And then additionally, like other amino acids that have hydroxyl groups, it is a major phosphorylation target. So a very frequent modification in the cellular environment of proteins is protein phosphorylation, typically through enzymes called kinases. And serine is a major target for that because it has this hydroxyl group. In fact, it's required for phosphorylation to occur. Additionally, another amino acid we discussed previously, tyrosine, can also be phosphorylated. Now, the next one is threonine, and this one has the letter T. And notice that this group also has a hydroxyl, except in this case, it's on the beta carbon, and then it also has another carbon attached to it, which is a methyl group. And notice that this carbon here, the beta carbon, is chiral, just like that of isoleucine, if you recall. And that's because it's attached to 4 unique groups. Additionally, just like serine and tyrosine, the hydroxyl group is a target for phosphorylation. The next one is asparagine, or N. And so now this is where it gets a little bit more tricky because most of the following amino acids that we'll talk about do not have an abbreviation that corresponds with the first letter of the name. So asparagine, what I'd like you to remember is that asparagine has N in the name, asparagine, okay? So that's how I typically remember that N is what denotes it. Now notice this side chain, it has this methylene group and then this carboxy group attached to an amino group. And this is called an amide functional group, all right? And this can actually form hydrogen bonds as well. And in some cases, it can undergo a reaction called deamidation. And when this undergoes deamidation, essentially this amide group is converted into a carboxy group, and it becomes another amino acid we're going to talk about called aspartic acid. Or if it's under basic conditions, it becomes the conjugate base of this called aspartate. Next, we have glutamine. And so glutamine has this weird abbreviation Q, okay? And really, this is actually an attempt to phonetically denote glutamine, because apparently the glue at the start can sometimes sound like a Q. So, you know, to make that work, I kind of try to pronounce this as glutamine, which is a little bit closer to a Q. But that's how I try to remember it. And notice that this one also has an amide side chain. It's basically like the asparagine N side chain, but with one extra carbon attached in it. And so this is also an amide side chain. And this is a very important molecule in the body because it's really important for essentially eliminating ammonia. And in fact, ammonia can be, ammonia is produced essentially as a byproduct of nitrogen waste in the body. And this can sort of act as a surrogate molecule of nitrogen waste. And eventually it can be converted into ammonia. And so the way it's carried in the bloodstream is via glutamine oftentimes. Now, this can also undergo deamidation similarly to asparagine, and it forms glutamic acid, or again, in basic conditions, the conjugate base, which is glutamate. Now, speaking of which, we now have the negatively charged / acidic amino acids because they have acidic and negatively charged side chains under more basic conditions. So the first one is aspartic acid. Now, this one is denoted by D. And this one's a little easier because if you pronounce it as aspartic acid with a DIC here, a DIC, then it's easier to remember that it's a D, aspartic acid, okay? And it has a methylene group and then this carboxyl group, so it has a carboxylate side chain. And normally under physiological pH, which is around 7.4, this is deprotonated. So it exists in this negatively charged state. And this is important for a variety of reasons because since it's ionic, it can form various electrostatic interactions, specifically ionic interactions. And then additionally, it can be used as for enzyme catalysis. Now, the next one is similar. It's glutamate or glutamic acid if we're talking about the acidic form. Okay, and this one has the letter E. So the reason historically this is E is because if you notice aspartic acid has D, okay, as the notation, and it only has one methyl group preceding the carboxyl group. But notice that glutamate or glutamic acid, it has kind of the same side chain, but with one extra methyl group or methylene group, CH2 group. And so the reason is because E comes after D in the alphabet, and since there's one additional carbon in the chain, they named glutamate E. So DE, one carbon in the chain, two carbons in the chain preceding the carboxyl group. So that's how you can remember it. Now, the other thing is with glutamate, it is actually a precursor molecule for neurotransmitters. And famously, glutamate itself is a neurotransmitter. So it's an excitatory neurotransmitter, very important in the central nervous system. And then also it can be used to synthesize GABA, which is an inhibitory neurotransmitter. So next, we have the final group, which are the positive or basic / alkaline amino acids. And so the first one of these is lysine. Now notice lysine has this long 4-carbon chain, and then it has an amino group. And normally under physiological pH, again, 7.4, this is actually protonated and hence carries a positive charge. And again, it's important for various ionic interactions in cells, therefore helping to dictate, in proteins, helping to dictate the structure of them. And then it can also be used for enzyme catalysis, similarly to the negatively charged amino acids. Now, again, this one is weird because it has K denoting it. Now, recall leucine is the one that already has taken the L, okay? So lysine, K and L are adjacent to each other in the alphabet, okay? Methionine actually, or M actually follows L in the alphabet, but since methionine has M, lysine is given K because that's the other letter that is next to L in the alphabet, all right? It's kind of a similar story as aspartic acid and glutamic acid, D and E. And then with leucine, since it's given L, we take an adjacent letter in the alphabet, in this case K, and give it to lysine, because lysine also has an L at the start of the name. And then we have arginine. So arginine is denoted by R, and again, this one's a little easier because you pronounce it pretty much as arginine. You know, R is pretty much what you hear at the start of the name, even though it starts with an A. So arginine is R. So this one has this, this, again, carbon chain, 3 carbons, and then it has this weird funky group, which is called a guanidinium group. Excuse me, I can't pronounce that, but guanidinium group. Okay, we're going to cut right here, and then I'm going to start again. So next we have arginine. So arginine, notice, is denoted by R, and this one's a little simpler to remember because when you pronounce arginine, it's almost like you're pronouncing it starting with an R. Arginine is R, okay? So it's an interesting side chain. Again, it has some carbons, and then it has this funky weird group here, NHC double bond to NH2 and a single bond to another NH2. So there's a bunch of nitrogens. And this is called, and I'll try my best to pronounce it, a guanidinium group. Guanidinium group, okay? And normally under physiological pH, it is also protonated and positively charged overall, all right? And again, that allows it to be present with enzyme catalysis, all right? And then importantly, this is a molecule that is commonly used to bind negatively charged groups. So a lot of proteins, for example, histone proteins, will have a lot of these positively charged lysines and arginines. And recall that histone proteins are responsible for binding to DNA double helices and helping to coil it or unravel it, hence making it accessible to various DNA replication and transcription machinery. All right, so histones are basically proteins that sort of help to organize DNA. And because DNA, the DNA backbone, it has those phosphate groups which are negatively charged, the way that those histone proteins bind is they have a lot of these positively charged residues, including arginine. And so that allows for ionic interactions to hold together the DNA and the histones. And these 2 amino acids are very important for that. And then lastly, we have histidine, which is very nicely denoted by H, okay? And this one, again, notice it has this ring here, and it's attached CH2 and then this interesting ring, okay? And it's called an imidazole ring. It has 2 nitrogens within the ring structure itself. And incidentally, this nitrogen here, okay, under physiological pH, it can be either protonated or deprotonated, all right? In fact, the pKa of really the proton that can bind to it and the bond that it forms with it is around 6. So it's really close to physiological pH, around 7.4. And that really means that since they're pretty close, there's an appreciable amount of this histidine side chain that is protonated. And this makes it a very convenient amino acid for something called acid-base catalysis, which is a specific type of catalytic mechanisms that enzymes employ. And you will see this frequently used for those sorts of catalytic mechanisms because it's able to essentially be a shuttle for protons and kind of exchange protons with other groups, perhaps in molecules that or substrates that the enzyme is catalyzing a reaction for. Okay, so now that we've talked about the amino acids, let's go ahead and try to do an MCAT style practice passage together. So here's the passage. Go ahead and try to read through it, analyze it, and then try to answer the questions on the right. And then we'll go through it together. Okay, so let's get into it. So it says researchers isolated a protein suspected of having distinct hydrophobic and hydrophilic domains. To test this hypothesis, the protein was solubilized in a solution containing water and a mild detergent. The solution was then passed through a hydrophobic interaction chromatography column, which binds hydrophobic regions of proteins. The protein eluted in 2 distinct fractions. Fraction 1 eluted with water alone, H2O, while fraction 2 required the addition of a high salt solution for elution. Further analysis of the fractions revealed the following. Fraction 1 was comprised of amino acids such as serine, threonine, and lysine, whereas fraction 2 was comprised of amino acids such as leucine, valine, and phenylalanine. So to confirm the structure, researchers then used X-ray crystallography and found that the protein folded into tertiary structure with hydrophobic residues buried in the core and hydrophilic residues on the surface. And then we have the questions. So before we jump into those, let's analyze this passage a little bit. So first they're mentioning some experimental analytical techniques. And the first one is this hydrophobic interaction chromatography. All right, so what we need to know about this is, in general, chromatography are kind of a group of techniques that are used for analysis in order to sort of, first of all, separate a solution of molecules or molecules in a solution. And they're separated based on certain properties. And inside this chromatography apparatus, in this case it's a column, there's going to be some sort of medium. And this medium has certain properties that allow it to variably interact with whatever molecules are passed through it. In this case, that medium seems to be hydrophobic or nonpolar, so it'll more strongly interact with other nonpolar things. So in this case, if you pass a solution of proteins, as in this case, through this chromatography column, the more nonpolar residues, proteins, etc., are going to interact with the medium more strongly than the less nonpolar ones, the more polar and ionic things. And generally speaking, the things that interact more weakly, less strongly with the medium are going to pass through the column more quickly and will be eluted first. And then those that interact more strongly will be retained for longer, and that's how they're separated. And then X-ray crystallography is simply a technique that's used to analyze structure of proteins. The specifics are beyond the scope of the MCAT, but just know that it's used for structural analysis. And then we see the two fractions that eluted. The first one that eluted was comprised of these polar amino acids, right? Because serine, threonine, and lysine are all polar, and lysine is actually charged and basic/ionic, okay? And then fraction 2 was comprised of leucine, valine, and phenylalanine, which are all nonpolar, all right? So that makes sense because these must have interacted more strongly with the hydrophobic column and therefore were retained for longer, and it took longer for them to be eluted. All right, so question 1. So it says amino acid properties based on the experimental results. This is the question. Based on the experimental results, which of the following best explains why fraction 1 eluted with water alone? All right, so answer choice A says fraction 1 contains hydrophobic amino acids, which readily dissolve in water. Well, for one thing, there's a clear inconsistency in the question because hydrophobic amino acids certainly do not readily dissolve in water. So we already know this is wrong, but then also it's wrong because fraction 1, we recognize these are all polar. So that's wrong. Now, B says fraction 1 contains hydrophilic amino acids, but do not strongly bind to the hydrophobic column. Now, this seems right because hydrophilic amino acids, these are hydrophilic, that's right, and those certainly do not strongly bind to the hydrophobic column. So this seems correct. Now, we could just stop there and say, B is the right answer and move on. And, that's okay to do, but I think when you're starting out, doing practice passages on the MCAT, answering questions, no matter how simple the question seems, I think it's always a good idea to practice going through each answer choice and ruling things out and just making sure, double checking. All right, so let's just practice that. So C, fraction 1 contains amino acids with aromatic side chains that interact weakly with the column. So for one thing, we see fraction 1 certainly doesn't have any aromatic side chain amino acids. I mean, none of these have aromatic side chains. And the other thing is that aromatic side chain amino acids generally are hydrophobic, so they would actually strongly interact with the column. So this one's wrong. And then fraction 1 contains acidic amino acids that are destabilized by the detergent. So, this one we got to think about because acidic amino acids, they are indeed polar, right? And these are all polar. But the issue is that lysine, the only charged one, right? This one is actually basic or alkaline. It's not acidic, right? And then these two are not acidic. They're not even ionized, all right? So that's why this one's wrong, okay? But we might have to think about that one a little bit before we rule that out. But we see that there's no mention of an acidic amino acid, so that's why that's wrong. So yes, B is indeed the correct answer, all right? So moving on now to question 2. So it says a mutation replaces a valine residue in the hydrophobic core of a protein with a serine residue. So the valine is changed to a serine. Which of the following is the most likely consequence of this mutation? So valine, right, V, is changed to serine, right, which is S. Okay? Now, this question is an example of why it's important to know something about the structures of these amino acids, right? And again, which group they belong to. Because here they're asking us, what is the effect of changing valine, which is a nonpolar amino acid, to a serine, which is a polar amino acid, which has a hydroxyl group, on the inside of a protein where everything is hydrophobic? So answer choice A says the protein's tertiary structure becomes more stable. So that seems wrong, right? That seems wrong because how would it become more stable if you have a bunch of hydrophobic interactions, right, which valine is a part of, but then you change that valine to a molecule that doesn't participate in hydrophobic interactions, which is serine, since it's polar. So that would actually do the opposite of what A is saying. It should destabilize it, theoretically. So then we see B, the protein will lose hydrophobic interactions, destabilizing the core. Well, that's actually exactly what we just said. So this seems right? Because again, we're changing an amino acid that's participating in those stabilizing interactions, the hydrophobic ones, to a polar one, which can't participate in them. So that would certainly weaken the overall stability of that core, that hydrophobic core, right? So, but again, we're going to check C and D as well. So C says the protein will gain ionic interactions in the hydrophobic core. Well, we know serine is not ionic, so it doesn't participate in anion, cation, or negative positive charge interactions. It's not ionic, so that's wrong. And then D says the mutation will have no effect on protein stability. Well, this one is certainly wrong, right? Because we've already established that most likely protein stability is going to suffer in this case, since we're actually weakening hydrophobic interactions. And, generally speaking on the MCAT, it's pretty rare to have an answer choice like D be correct, where the answer is like nothing happens, or none of the above or something like that. That's generally the wrong answer. But in this case, we can certainly prove it, okay? So then question 3 says, based on the following amino acids, which is most likely found on the surface of the protein? So for this question, even without looking at the answer choices, we got to realize, right, they're talking about the surface of a protein. And usually proteins, whenever we're discussing protein structure, we're thinking about it in the context of like an aqueous solution similar to the inside of a cell. And so there's water all around, and we know that generally in the way that proteins fold, based on the characteristics of their amino acid constituent side chains, right, they want to make sure that those hydrophobic side chains are away from those water molecules. So they get packed into that core, that hydrophobic core, and then they are shielded from the outside aqueous solution from those water molecules by polar amino acids and ionic amino acids that then group on the outside of that core. They form sort of a shell structure, right, protecting that core. So they're asking us which of the following would most likely be found on the surface of that protein amongst that shell. So we know conceptually it needs to be something that's hydrophilic or polar. All right, so we're just going to rule out the non-polar ones, and it's that easy, right? Or just identify the one that's definitely polar. So number 1, A, glycine. Well, we know that's wrong. That's non-polar. Then B, arginine. Well, look at that. Arginine, we know it has an amide group. It's polar. So that's got to be right. But then look, phenylalanine, aromatic side chain, non-polar. Valine, also non-polar. And then alanine, it's just a methyl group, non-polar. And then boom, that's it. Arginine is the only polar amino acid here. So that's got to be the right answer. That could be the only one on the surface. Okay. And by the way, quickly, just practice assigning the 1-letter abbreviation to each of these, okay? Because that's an important skill to have, because it's very possible they'll use these interchangeably. I mean, it's just that they could have just given you these amino acids as letters, and you would have had to recognize that G is glycine. Arginine, remember, is N because of that N right there. That's how I remember it. Phenylalanine is F because phonetically it starts with F, phenylalanine. Valine is easy, it's V. And so is alanine, it's A, okay? So, whenever, especially starting out, if you're unfamiliar with this topic, try to always, whenever you see a name of an amino acid, recall the 1-letter abbreviation for it. So that's our video on amino acids. Check out our website at mcatsimplified.com.